So I want to look at this. All right, so this is what I sent out with the learning outcomes. I'm going to kind of try and I'll put me down here at the bottom, maybe. All right, so now we ended up with the endocrine system, and I just wanted to let you know the endocrine system will not be a big part of the final. It'll be one of the chapters where I'll probably limit the questions on. The big thing is, simple as knowing the difference between endocrine and exocrine. Right? Endocrine glands do not have a duct where the exocrine glands do. And obviously, you better know that the endocrine glands are the ones that make up the endocrine system. Do want you to know the difference between steroid and non-steroid hormones. Again, steroidal hormones are lipid soluble, non-steroidal are water soluble, and they have a big difference in the way they affect the target cell. Now remember, lipids, which are the steroid hormones, can move through cell membranes. So they go directly to the nucleus and cause the creation of a new protein or enzyme, where non-steroidal hormones, because they're water soluble, they cannot go through without help, without the cell membrane, without help. And so they bind to receptors on the cell surface, and through a chain of events cause already created enzymes to become activated. All right, so that's a big difference between the two. The next really on this, and just to give you a little heads up, I'm trying to hit the highlights. I'm not saying that the things that I don't go over, are just completely off limits. I wouldn't, ask you anything that I thought was really in depth that I'm not going over, but understand that you're responsible for everything. I'm trying to give you an idea of where to guide your studies for this. Now, knowing the major endocrine glands and the hormones they secrete is a big thing. So I do want you to know that. I want you to know what endocrine glands secrete what hormones. You don't have to know the actions of all the hormones, but you do need to know what glands secrete which hormone. Now, I'm going to tell you, if I see a question that's going to involve calcitonin and parathyroid and how it regulates blood calcium levels, that'll probably be one I would pick on as far as action goes, as well as insulin and glucagon. Those are two sets of homeostasis processes that I do believe you should know. So then we go to blood. So again, now we get to the part where we're looking at things that seem like 10 years ago. All right. So blood is made up of two distinct sections. One is called plasma, which is the liquid portion, and it's got small molecules, and then one is called formed elements. In the formed elements, there are three types of cells, and you need to know both the common name and the scientific name. So red blood cells are called erythrocytes, white blood cells are called leukocytes, and the platelets are called thrombocytes. Those make up what are called the formed elements. And the red blood cells, or erythrocytes, are by far the most common portion of the formed elements. Now, red blood cells are going to be closely monitored by the body, because after about 120 days, they no longer carry oxygen efficiently, so we get rid of those and produce new ones. It is going to be controlled by a hormone called erythropoietin, right? That's going to be generally released by the kidneys. The spleen can do it as well, and whatever. And it's going to go and it's going to cause creation of red blood cells. Understand that all of the blood cells, all the formed elements, their precursor cell, which we call the stem cell, hemopoietic stem cell, is going to be found in the red bone marrow. You do need to know that. So hematopoiesis is the general catch-all name for any type of blood cell. Erythropoiesis is for specifically red blood cells. Now, in the white blood cells, there's 5 different types of white blood cells. Now, the blood cells are divided up into granulocytes and agranulocytes. There are 3 granulocytes and 2 agranulocytes. So the 3 granulocytes, the very first one's called a neutrophil. It is the most common white blood cell by far. 60% of all the white blood cells are what they refer to as a neutrophil. The things I want you to know about a neutrophil: one, first and foremost, is it is a phagocytic cell. It is an eater. The other thing I want you to know is that it has a multi-lobed, weird-shaped nucleus. You do not have to know its original name, polymorpholeucocyte, polymorphonucleocyte, which means multi-lobed, weird-shaped nucleus, and that's cell. So you don't have to know that, but that's the gist of it. The next one is called the eosinophil. Eosinophils stain red. And then we had the basophils. The basophils stain blue. Now, I want you to know that basophils have histamine and heparin granules. Now, eosinophils, and when we get in the lymphatic chapter, I do want you to know that eosinophils are what are associated with allergies. All right. Now, neutrophils are the most common. The eosinophil and basophils are the two least common. That is the section that we call granulocytes. Agranulocytes consists of lymphocytes and then monocytes. Now, lymphocytes are the grand poobah of the cells, in my opinion. And in fact, outside of a neuron, they are just absolutely the most amazing cell. They are what we look at as our immune system. We have T and B cells, we'll learn, and there are types of lymphocytes. Then we have the monocyte. What I want you to know about the monocyte is it is not only the largest of the leukocytes, but it is also as a phagocytic cell. So neutrophils and monocytes are phagocytic cells. They're big, and the monocyte can leave the circulation and live outside of it. And if it does, it is then called a macrophage. And macrophage literally means big eater. And so that's what it is. And it has a kidney bean shaped nucleus. Now, outside of that, I'm going to be very brief on any questions on, too much into these. But the next section is going to be blood typing. There's going to be a couple questions on blood typing. Remember that first and foremost, all of our cells are going to have little markers on them that tell us that it is us. And that is, we're going to refer to this in blood typing as an antigen. We'll learn in the immune system that antigen specifically refers to something that's an invader. But in here, antigen we're going to look at is what our body's reading. And there's only 3 antigens: an A antigen, a B antigen, and an RH antigen. A and B are looked together in what we call the ABO system. I'm going to look at that separate from the RH system. But again, there's only 3, period, no more. So if we separate the A and B out, they act differently than the RH, because automatically, as my body is developing as an embryo, once one of these dedicated pathways happens with my body, it's going to be a combination or some sort of the AB, all right? So I can have only an A antigen, which would mean I'm type A. I can have only a B antigen, which means I'd be type B. I can have both of them, which means I'd be type AB, or I can have neither, which means I'd be type O. Those are the only 4 types of the blood types on the ABO system. Now, with that, whatever antigen I do not have, my body will produce an antibody that is whole point in life is to hunt down and kill that antigen. So let's say I'm type A, which means I have A antigens on my cell. I do not have type B. Type B is my enemy. So my body makes antibodies that are called anti-B antibodies, because their whole job in life is to look for B antigens and attack it. So if I'm type B, that means I have an B antigen. I do not have an A antigen. So I make anti-A antibodies. If I'm type AB, that means that I have both of them. That means I lack neither one. So in type AB, I have no antibodies. And then if I'm type O, that means I have neither one of them, neither A or B. I should say nor, right? Neither A nor B. And so because I don't have either one of them, I'm going to have both antibodies, anti-A and anti-B. So when we're looking at blood typing, we're named for the antigen. Now, if I am trying to donate blood, I want to think about my antigen and the other person's antibody. So if I'm type A and I want to see if I can donate to someone, I know that I have A antigens because I'm type A. If they have anti-A antibodies, I cannot give it to them. So type B cannot receive blood. I cannot, if I'm A, I cannot donate blood to type B because they have anti-A antibodies. I'm not going to go through the whole thing on that, right? If I am type B, right, I have B antigens. I cannot give to anybody with anti-B antibodies, right? And the two types that have anti-B antibodies are A and O. Now, receiving is, then I'm looking at my antibodies. I'm not worried about my antigens. The antigens I'm worried about are the ones I'm putting into my body. And the only reason I'm worried about them is if my antigens don't, or if my antibodies don't like that antigen, it's going to attack it. So if I am type A, which means I don't have B antigens on my cell, which means that I have anti-B antibodies, any cell that has a B antigen cannot come in my system. All right, it's as simple as that. Now, when you go through and look at all of this, you're going to see two things special about AB and the other thing special about O. If I'm type AB, that means I have no antibodies. Because I have no antibodies, I can put any cell into my system because I have nothing that would attack it. So type AB is considered the universal recipient. If I look at type O, because they have no antigens on their cell, I can put my cell into anybody's body because since I have no antigens, I don't have to worry about any antibodies. So type O is the universal donor. All right, now again, I can't really spend a lot of time going into that. I'm trying to get this set for you to release it so that you got something to study with. So the cardiovascular system, we continue on. We start talking about the heart and the blood vessels. You need to know the circulations, right? We all learn the systemic circulation when we are in high school and whatever, general biology, general science. And that's where we get the messed up information that all arteries carry oxygenated blood and all veins carry deoxygenated blood, because that's the way it is in the systemic circuit. The systemic circuit is oxygenated blood leaving the heart, going out to the tissues, dropping off oxygen, picking up carbon dioxide, and coming back to the heart. There is another circulation called pulmonary circulation that leaves the heart carrying deoxygenated blood to the lungs, where it will drop off the carbon dioxide, pick up the oxygen, and come back to the heart. So that's the pulmonary circuit. You got to know both of them. And we also talked about the coronary circuit that's going to go from the heart back to the heart muscle, and then through the coronary sinus comes back to the right atrium. Now, I'm not worried about the location of the heart. I hope you know it's the mediastinum. If I see that as a question, I'll probably pick it because if I can, because that should be an easy one. I'm not worried about the layers of the heart. Now, the function, identify the major parts of the heart, this little guy right here, and the discussion of the function of each part. Look, so we're going to go through, and the next one is outline the pathway. So I'm going to go through the pathway of the heart and describe things as we go along. All right, so I've got deoxygenated blood coming back to the heart through the superior and inferior vena cava. Superior vena cava draining blood from the upper extremities, inferior from the lower extremities. Comes back to the right atrium. The heart's made up of two sets of chambers. Atrium are the entryway, ventricles are the pumps. So it's coming into the right atrium. Now the right atrium is dealing with deoxygenated blood. It's going to go through the tricuspid valve from the right atrium into the right ventricle. Now the tricuspid valve is a one-way entryway into the ventricle. When the ventricle contracts, which is called systole, it is going to squeeze. It increases the pressure and it's trying to squeeze the blood out. When it does that, the tricuspid valve will close. It closes by way of a mechanism that starts with finger-like muscles called papillary muscles that are attached to cord-like tendons called chordae tendineae. Those attach to the valve and keep it from being flung back. So the tricuspid valve between the right atrium and the right ventricle, when the right ventricle squeezes, called systole, it's going to push blood out, and since we're talking about deoxygenated blood, we're trying to get it to the lungs. So it pushes it out through the pulmonary semilunar valve into the pulmonary trunk. Now, the pulmonary trunk is an artery because it's going away from the heart. It doesn't matter that it's carrying deoxygenated blood. We're talking about the pulmonary circuit right now. So the pulmonary trunk divides to pulmonary arteries. They go to the lungs. There we get the magic of the respiratory system that we looked at. The respiratory membrane, as the blood goes across, we're going to have carbon dioxide, which is high in the blood, jump across into the alveoli, into the air, and then the oxygen, which is high in the air, jump across into the blood. Now the blood's going to come back to the heart. It's going to start with the left atrium. It's going to come back by way of the pulmonary veins. In the vein, these pulmonary veins carry oxygenated blood. Comes to the left atrium. The left atrium, from there, it's going to go into the left ventricle, and it's going to pass a valve now with only two flaps called the bicuspid, but it is also called the mitral valve, and you need to know both of those. It has the same makeup with the chordae tendineae and papillary muscles as before. So in this, I'll explain that when the ventricle relaxes, as it opens up, there's a suction, and it pulls the blood in from the atrium. That is called diastole. Now, when it goes into systole or contraction and increases the pressure, the bicuspid or mitral valve slams shut, and the blood gets ejected into, this time, the aorta. So it's going through the aortic semilunar valve to get into the aorta. From the aorta, it goes out to the body, does its magic, drops off oxygen, picks up carbon dioxide, and comes back in through either the inferior or superior vena cava. The whole cycle continues. Now, when we look at how the blood vessels work, there's a difference between arteries and veins. They both have the same three layers: an inner tunic, a tunica internica, tunica media, tunica externa, but they're very different. In arteries, there's going to be a much thicker tunica media because of the smooth muscle that helps control the blood. In veins, they have a much thinner tunica media, but along the tunica interna, we have valves, and they're basically semilunar valves, and they help the blood flow in one direction because arteries have a lot of pressure in it, pushing blood in one direction. Veins do not. And so they rely on skeletal muscle pumps and respiratory pumps to push the blood back to the heart. And so those are the difference between those. Now, in the capillaries, and it's not discussed on here, but you do need to know that it is on the arterial end of a capillary. It's the precapillary sphincters that help regulate how much blood gets into the capillary bed, and that it is filtration because of the hydrostatic pressure that pushes the fluid out. The fluid is drawn back into the capillaries on the venous side by way of osmosis. Now, there is a small percentage of fluid that is pushed out that doesn't come back in. That's called tissue fluid, and it builds pressure. That's what we look at in the lymphatic system. That buildup of tissue fluid will push into the lymphatic system, and now it's called lymph. Now, blood pressure is different in the arteries and the veins. Blood pressure it drops as it gets to the capillaries. Once it goes through the capillaries, it is almost non-existent. And so that's why it requires the skeletal muscle pump and respiratory pump to push the blood back to the heart. Now, comparing pulmonary and systemic circuits is what I went into before. Pulmonary is going to be carrying deoxygenated blood to the lungs and then carrying oxygenated blood back. In the pulmonary circuit, it is opposite. The arteries carry deoxygenated blood, the veins carry deoxygenated blood. Remember, if you want to make that a true statement, it is always that arteries always carry blood away from the heart. Veins always carry blood to the heart, right? Whew. All right, so the lymphatic system. Now, first of all, understand that there's it's just basically the bottom third of the lymphatic system that gets really complicated with the immune system. And so it's going to be just a portion of this. So there is going to be things in here that you'll have to identify and understand, not identify as in a picture, but understand. You need to know, as it says, the first highlighted one, tissue fluid, right? Plasma gets pushed out in the capillaries. At that point, it's called tissue fluid. If it comes back into the capillaries, it's back to being called plasma. But if it moves into the lymphatic system, at that point, it's called lymph. Lymph is designed to drain the tissues. And then as it goes through, it's going to be filtered by things called lymphatic nodules or lymph nodes, and those will help signal our body if we have an infection. Now, on this, there's one that's not highlighted, discuss the locations and functions of the thymus and the spleen. You do need to know a little bit about those two, but it's nothing major. I mean, I doubt that I'm going to ask a question about it, but you know, the thymus is where T cells go to be mature, and then the spleen is basically a lymph node for the blood. The spleen is the largest lymphatic organ. Now, when we get into immune system, distinguish between innate, nonspecific, and adaptive, specific defenses. Innate defenses are always they attack everything. Whatever's trying to get in us, it's going to attack it the same way. It's quick. Where the adaptive is and that's why it's called specific, will only attack things that it's been trained for. And it'll get trained for one particular thing, and that's it. Explain how the two major types of lymphocytes are formed and activated, and how they function immune mechanisms. This is kind of a big one. T and B cells, they're both called lymphocytes, but T and B cells, they're both formed from the red bone marrow, but the T cells leave early and go and mature in the thymus. B cells stay in the bone marrow and mature there. T cells are what we call cellular immunity, because they go and they will literally fight cell to cell, where B cells produce antibodies, and those antibodies flow through tissue fluid, through the plasma and tissue fluid and lymph or whatever. So they use the blood, they use the fluid of the body, and fluid of the body, the old school name for fluid of the body is humor. So B cells are what are considered the humoral defense. Now, both of them will produce memory cells. Once I have come in contact with a certain antigen, a pathogen, my body will produce T and B cells for those. As those T and B cells as the body recognizes there's a pathogen, the T and B cells will become activated. T cells will activate into helper cells and cytotoxic T cells. Helper cells are the ones that are going through and kind of recruiting the B cells to become active and getting everything to work together. The cytotoxic T cells go straight to the source, and they fight it. Now, B cells, when they become active, they're going to produce memory cells. T cells do that too, that hang back. But their activated cells are called plasma cells. Plasma cells are what produce antibodies. The antibodies flow through the tissue fluid, and then they are going to be what comes back and attacks through the system. Now, there are a couple of different types of antibodies, and you need to know them. There's 5 types. You need to know that the most common is called IgG. So the 5 types are IgG, IgA, IgM, IgD, and IgE. And I told you that if you kind of invert the last 2, you can remember GAMED, G-A-M-E-D. Those are the 5 types of antibodies. Now, again, the main thing is I want you to know IgG is the most common type. I would like you to know IgE is what is responsible for our allergies. Now, on here it talks about the difference between active and passive immunity. And active immunity is when our body does something to produce the antibodies or the memory cells. Passive immunity is when it comes through, it comes from an outside source, right? Again, trying to get this, I didn't set the timer. I'm trying to make sure I don't end up talking too long on this. I don't want it to be 3 hours. The digestive system. The digestive system is going to be what's breaking down the food we give it so that it gets to the component parts. It has to get to the basic parts in order for them to be absorbed. Well, that's called catabolism. Then when our body absorbs it, it takes it around and it builds you, which is called anabolism, right? So then we have certain structures along the digestive system, the digestive tract, that are going to have different parts along it. So the whole thing's called the alimentary canal. And then we're going to have things like salivary glands. And then we're going to see a part just in the first part of the small intestines where we got the gallbladder, liver, and the pancreas come together. So in that, the salivary glands produce saliva. There are 3 salivary glands: the parotid around the ear, the submandibular underneath the mandible, and sublingual underneath the tongue. Not going to get into much about those, but when you go, you chew, you do need to know that the only place that we go through mechanical digestion is in the mouth and chewing. As we swallow, it goes through the esophagus, which has no digestive enzymes that are produced there. The esophagus, through peristalsis, pushes the food, which is called chyme at that, or called bolus at that time, gets into the stomach. Once it's in the stomach, it's going to start mixing with what we call gastric juice. At that point, it's called chyme. The gastric juice there, the 2 main ones, know that the chief cells produce pepsin. It's always produced as an inactive form called pepsinogen. All protein enzymes have to be stored and released as an inactive form, right? Or it'll destroy its container. So pepsinogen, which turns into pepsin, is released by the chief cells, and then the parietal cells produce hydrochloric acid. Now, this mixes up. At this point, it's called chyme, and then little by little, as it enters into the duodenum, the first section of the small intestines. Remember, duodenum, jejunum, ileum, three layers of the small intestines. So it enters into the duodenum. At this point, we're going to have the big 3: the liver, which does a lot of stuff, but in this, we're just looking at the fact that its breakdown of red blood cells is producing bile, and the bile gets stored in the gallbladder. So I've got the common hepatic duct, the duct coming out of the liver, the cystic duct, the duct coming out of the gallbladder, joining together to form the common bile duct. Then we're going to have the pancreas, which the pancreas is the only organ in the body that has both major endocrine and exocrine portions. The exocrine portion that we're looking at in this is the pancreatic juice, and it's going to be secreted onto a duct that's exocrine, and it's called the pancreatic duct. Now, from the endocrine side, remember that the 2 things is glucagon and insulin. Now, the pancreatic juice comes out, and that's all going to go in the duodenum. Now, in the duodenum, there's going to be a special type of motion called segmentation. Remember that peristalsis is simply moving things from the beginning to the end. But in the small intestines, we're going to have this thing called segmentation, where the small intestines will contract on either side of the chyme and then kind of mix it all together so that all of these enzymes are mixed in. As the enzymes do their work and start breaking down the particles, remember the big 4, and really I'll tell you the big 3 of the organic molecules. Organic molecules, the 4 of them are carbohydrates, lipids, proteins, and nucleic acids. Now, nucleic acids, the building blocks are nucleotides. I doubt I'm going to get into that, but I could. The other 3 are the ones that, since I've got limited questions, are the ones I'll probably concentrate on, and that's kind of the next chapter. But carbohydrates, the building blocks, are called monosaccharides. The main monosaccharide is something called glucose. Lipids are made up, the dietary lipid or triglycerides. They're going to be made up of 3 fatty acids. And remember, the fatty acids are either going to be saturated or unsaturated. Saturated fatty acids have a hydrogen at every spot, and they are basically animal in origin and will be solid at room temperature, like butter. If we're talking about unsaturated fatty acids, they will be liquid at room temperature, and they will be generally from plants, vegetable oil. All right. So, and then proteins are made up of amino acids. Now remember, amino acids, amines, are bound together by peptide bonds. That's why if we've taken a protein, we've unraveled it and start sectioning it off, they're peptides. Peptidases are big things in that. Now, from the small intestines is where the absorption is basically going to happen. The brush border cells are where the enzymes are going to do the official final breakdown of the the final components, absorb them. Now you need to know that all of it gets absorbed into the circulatory system except for one. There is the lipids have to go through the lymphatic system first. There is a special lymphatic capillary called a lacteal that is found in the small intestines, and it is simply for absorbing dietary lipids. Now, once it goes through the small intestines, it's going to go through the large intestines, where most of it is just about absorbing water. There will be some vitamins, some smaller things that get done there. I am so sorry. Whew, that's bad. But its main thing is it's absorbing water. At this point, it is called feces, and then we will get rid of the feces. Now, understand that sympathetic and parasympathetic, two divisions of the autonomic nervous system that are controlling things. Sympathetic is going to be inhibitory to the digestive system. Parasympathetic is stimulatory. When you get it, that's why, again, if you're driving down the road and like on Farrell Parkway, and the speed limit there is like 4, and you see a police officer, you have the sinking feeling in your stomach. It's the sympathetic nervous system switching on, which is shutting off your digestive system. All right. And again, remember, parasympathetic in here is mainly the vagus nerve, cranial nerve 10. So then we move on to the next chapter, which was nutrition and metabolism, basically the nutrition of the building blocks of it, like we went over. Know about vitamins and minerals, know a little bit about those. Know, A, D, E, and K are all lipid soluble vitamins. That's why you can put vitamin E on your skin and it's absorbed. B and C are water soluble vitamins. They don't absorb through your skin. And again, it goes back to what the characteristics of something being a lipid is. The respiratory system. In the respiratory system, you need to know general functions of it, some parts of it. I would know the difference between the upper and lower respiratory system. If it doesn't matter about the larynx, if the larynx is in there, don't use that as the key. But the upper respiratory system is anything above the larynx. So if it says pharynx, nasal cavity, whatever, that's upper respiratory. Anything below the larynx is lower respiratory, trachea, bronchial tubes, whatever. Inspiration, expiration, breathing in and breathing out is all about changing the pressure inside our thoracic cavity. It's mainly done with the diaphragm. When the diaphragm is stimulated, it flattens out, causing a suction to happen because it's decreasing the pressure inside the lungs, and it's doing it by increasing the volume. Boyle's law. Doubt that I'm going to get into that, but you probably should know that still. Now, if I need to help breathing in or breathing out, like let's say I need to cough or sneeze, I'm going to recruit intercostal muscles. If I'm breathing out, the intercostal muscle is going to be the ones on the inside. So they're internal intercostals, because what I'm trying to do is squeeze my lungs together, squeeze my rib cage in. If I'm breathing in like I just did, it's the external intercostals, because they're trying to take my ribs and flare them out. That allows for more air to come in. We talk about the respiratory volumes and capacities. Really, the big ones are going to be on the volumes. I'll go over them just really quickly. Remember that lungs have a certain amount of air that can come into it. It's roughly 6,000 milliliters. It's going to be divided up into 3 volumes. The first one is right in the middle. It's the most obvious one. It is breathing. It's your normal breathing volume, and it's called tidal volume. For the average person, it's 500 milliliters. Now, I'm going to have a section of empty space that I could put air in above that, and that is called inspiratory reserve. And then at the bottom of a normal breath, I still have air left in that I can push out, which is called expiratory reserve. So those are the 3, and those 3 I have access to, and that's why they're called vital capacities. The 4th one I do not have access to. It is a leftover. If I breathed out all I could, what was left would be an amount of air that's stuck in my pulmonary tree. And so that is called the residual volume. So the 4 volumes are tidal volume, which is normal, 500 milliliters. The only one of the volumes I want you to know the number. I have two sets of reserves, inspiratory and expiratory reserves, for if I need to breathe in extra or breathe out extra, they're there. And then the amount of air I'll never be able to breathe out, called the residual volume. Those are the 4 volumes. Now, the capacities are divided up, and there's 4 of those also. One of them is pretty straightforward. It's called total lung capacity. That's just adding all of them together. It's roughly 6,000 milliliters. Then I have vital capacity. Like I said, those are the ones that I can use. That's the 2 reserve tanks and tidal volume. Then the other 2 are the dividing line at the bottom of a normal breath. I have inspiratory capacity, which is after I breathe out all the way, how much room do I have to breathe in? So it is tidal volume and inspiratory reserve volume. Then if I drew a line there and I looked at how much air was left over, I'd have some air that I could use and some that I couldn't. So I would simply call it the functional residual capacity. And that's the one that throws people. But again, make sure you know the volumes for sure, the 4 volumes. Now, controlling breathing is going to be done in the brainstem. Now, we've got 2 areas, the medulla oblongata, which is where the main area is, and the pons. The medulla oblongata has a front section and a back section that are for respiratory area. The ventral or anterior area is for normal breathing. It is what's out in front. It's my normal breathing. Just breathing normally. It's controlling my tidal volume. If something happens and I know I need to breathe in deep, like just now when I knew I was getting ready to talk a lot, breathe in deep, it's the dorsal group. The dorsal group is going to modify that normal breathing and help me know when I need to breathe in extra or breathe out extra. And then finally, the pontine group, the one in the pons, sets the upper limit. It is going to be the one that tells my body when I need to, I can't breathe in anymore. So if I try to breathe in, I'm going to have the pons set the upper limit. That's one of the reasons that I like to remember it as the pons is above the medulla. So it sets the upper limit. Now, how the blood transports oxygen and carbon dioxide is an important thing. Carbon dioxide first, or oxygen first. Oxygen is mainly carried by hemoglobin. There's a small percentage of oxygen that's diffused into the plasma, but the main way is through hemoglobin. Now, carbon dioxide has 3 different ways. Some of it is dissolved in the plasma. Some of it is bound to hemoglobin, but the majority of it goes through that little thing that we looked at with the bicarbonate buffer system, right, and how it works. Now on here, I'm not set up to draw on it right now. I'm trying to record this while I can because I've got to get busy trying to make these tests. This one and my 210 final is kind of tough. So I don't have time to draw this out and figure out how to do it, but you need to look into that, right? Now discuss gas exchanges in pulmonary systemic circuits. Again, need to know respiratory membrane in the pulmonary circuits, 4 layers, the 2 layers from the capillary, 2 layers from the villi, and it has to be very thin. In the pulmonary circuit, we're having deoxygenated blood become oxygenated. In the systemic circuit, we have oxygenated blood becoming deoxygenated. Now, the urinary system, now we're getting into things that are relatively recent. You need to know the organs of it, the 2 kidneys, the ureters, the urinary bladder, and the urethra. You need to know the major parts and functions of the nephron. So the nephron's the functional portion of the kidneys. This is what is going to be doing the work. Now, in the nephron, you're going to have 2 main parts. You're going to have the corpuscle and then the tubule. And so the corpuscle has the glomerulus, which is the specialized little capillary bed in it. And then the corpuscle is the little capsule, or the capsule is the part around it that catches the filtrate. All right, so we're going to have an afferent arteriole coming into the glomerulus and an efferent arteriole leaving. That efferent arteriole will turn into what's called the peritubular capillary bed. Now, the tubule is going to have 3 parts. proximal convoluted tubule, nephron loop, distal convoluted tubule. And that paratubular capillary bed is going to hug all of those. And it's going to have a lot of give and take. So we're going to have things that have been filtered out that our body doesn't want to lose. And so they get reabsorbed back into the blood. That's called tubular reabsorption. Then there are things in the blood that we need to get out into the filtrate, and that's called tubular secretion. Now, our kidneys, you need to know that roughly 180 liters every 24 hours gets filtered through our kidneys, and the majority of that is going to be reabsorbed. The majority of stuff happens in the proximal convoluted tubule. The nephron loop is what's going to control the concentration, or control the volume, I should say. And so as we're going through, the distal convoluted tubule is what we're going to do the fine tuning. That's where homeostasis can kind of kick in, where it's going to let, the body, you know, modify things if the pH is out of alignment, if calcium's out of alignment, stuff like that. That's going to be a big part of it. So the nephron, after it does that, it's going to connect to a collecting duct. And the collecting duct is where it's going to drain what will be eventually called urine. Now, the collecting duct is going to be it's going to drain into a tubing system. Now, before I get to the tubing system, if I go back to the macro view of a kidney, the macro view is I've got a renal cortex, and then I've got a renal medulla. All right, the renal cortex is the outer layer, and that's where the majority of the nephrons found. The renal medulla is where we're going to find these things called renal pyramids. In between renal pyramids are renal columns. The renal pyramids are where the nephron loop is going to be dipping into, and that's where the collecting ducts go through. So if I'm looking at it, it's kind of an upside down pyramid. Those collecting ducts, as they come down, they're going to drain into what's called a minor calyx. Every renal pyramid is going to have a minor calyx at the point of it. So the fluid, which is now called urine, goes through a minor calyx. The minor calyxes will join together to form major calyxes. All of the major calyxes funnel into an area called the renal pelvis. The renal pelvis then drains out, and as it leaves, the renal pelvis turns into ureter. All right. And the ureters are going to drain the urine into the urinary bladder, and then the urinary bladder is going to store it until we have a certain amount of volume, and then it'll go through the urethra. Now, the urinary bladder is lined with transitional epithelium, and inside the urinary bladder, there's going to be this thing called a trigon. And that's just simply an area that looks like a triangle, and it's only because the transitional epithelium is not going to be found there, so that we don't have occlusion of the two ureters and the one urethra. All right. This next chapter won't be a big one, but I do expect you to know pH and how both the respiratory and urinary systems regulate it. You need to know what our normal body pH is. You need, and it's 7.4. It's a range, 7.35 to 7.45. That is what's referred to as physiological pH. And know that our body is constantly trying to work on that. If it's a problem with, if it gets too acidic, it is called acidosis. If it gets too basic, it is called alkalosis, because again, in the body, any basic part is called alkaline. If one of those happens and it's because of, say, the lungs, I have a respiratory issue, it is called respiratory acidosis or alkalosis. If it's because of a kidney issue, it is called metabolic alkalosis or acidosis. Now, going into the reproductive system, which is the last one here, I just want to tell you it is basic stuff. This is not going to be anything major. This is mainly stuff you looked at in lab. All right, the difference between mitosis and meiosis. I know you looked at some of the meiosis. You need to know that creating sperm is called spermatogenesis. Creating eggs or ova are called oogenesis. That is what males' meiosis is referred to and females' meiosis is referred to. Now, the basic structures and functions of both of these, are going to be the same basic structures. Testes Our testicles are the male reproductive organs and our glands, and then the females are our ovaries. Now, you know, as you go through, I'll actually also send the link to the lab portion of it, which I know you went over, but I'm not going to ask anything that's not on there, except for one exception. I hope everyone here knows that testosterone is the male hormone, the male sex hormone, and estrogen and progesterone are the females. The male testosterone is produced by the testes, the ovaries produce estrogen and progesterone. So I hope that that helps. Again, I wish I could spend a ton more time on it. I feel bad. I hope that this gives you a little idea.